So please interrupt me anytime you have any questions. So we just want to make the training a little bit casual. Right? So the first part will be about the Illumian sequencing overview.
Do you have any days left on? Do you have any handouts for us to find?
I will pass through all those PowerPoints to you guys after the training.
I captured it.
Okay. So this session we'll go through the major steps in Illumian sequencing workflow, and then I'll discuss the cluster generation process and the sequencing process, which we make use of the technology called sequencing by synthesis. So basically, the whole Illumian sequencing workflow can be divided into 4 major parts. It starts from library preparation. So I guess some of you should have already familiar with this part, as I know you have sent out samples for sequencing. So library preparation, we have lots of different strategies to prepare your library depending on your application. So after the library is generated, the first part that comes across with the instrument is to form the cluster, which we call the cluster generation. And in NextSeeds, the cluster generation is inbuilt. So that means you don't have to purchase another additional instruments for doing the cluster generation. So after you load the libraries to the NextSeeds system, the first step that we're going to do is the cluster generation. And after cluster generation, and it will automatically proceed for the sequencing. So, and then finally it will do the primary data analysis. So the output file after the sequencing is the base code files. So unlike the MySeq, we have an inbuilt secondary analysis software called MySeq Reporter, and the output file that you will get is the FastQ files. So it's more similar to the HiSeq system. So what you get is the base code files, the BCL file, and you have to use the software to convert the base code files to FastQ files. All right, so let me first introduce the library preparation. So let's do a quick survey. So how many of you have experience in preparing libraries? 1, 2, 3. Okay, so good. We have some experienced users. So this is basically a structure of a library. So when we talk about library, actually what we want to do is to add an adapters to the ends of the DNA insert. So the DNA insert is your target of interest. So it could be depending on your workflow. So if it's DNA sequencing, of course it's DNA. And if it's RNA, because with library preparation you have already converted the RNA into cDNA, so it's still double-stranded. And what we call the abductors at the ends of the DNA, actually we can subdivide the abductors into 3 different parts. So the first part would be the P5 and P7 sequence. So 5' P5 and 3' P7. These are the complementary sequence to the oligos that we coded on the flow cell. And then we will also have the read-one sequencing primer binding site and read-two sequencing primer binding site. So just like what you do in the standard sequencing, you have to have a sequencing primer, right? So this is the primer binding sites. And the third part is the index, or some people call it barcode. So you want to be multiplexed, you want to put a lot of samples in one single run, and that's what we manually introduce a unique sequence for different samples. So it could be around 6 to 8 nucleotide lengths. So each sample will have this unique index sequence, and after sequencing, you will use the unique sequence to do the demultiplexing. Right? Any questions so far? And how we prepare the library, we have different types of methods. Like the ligation, you can ligate the adapters to the ends of your DNA insert, or you can use PCR to incorporate the index. So different commercial kits uses different methods. So all can do. Alright, so after preparation of the libraries, the next step is the cluster generation. So what is cluster generation? So this is a typical flow cell. So this is a flow cell for the HiSeq, but the concept is the same. So actually, when we magnify the flow cell, you can see there are many pre-coded oligos on the flow cell. Of course, it's random. It's not essentially like this in a very regular way. So it's random coded with both complementary P5 and P7 primers. And what we do is, so when we denatured and diluted the library and then loaded it to the instrument, the single-stranded DNA library will bind to the flow cell. So it will bind to a complementary sequence because we have P7 and P5 sequence at the end, right? So what we do is want to amplify this single template into a clone so that we have enough sensitivity for the cameras to detect the signal, right? So what we do is to try to amplify this and to around 1 to 2 microns of each cluster. So this is a picture of the cluster they captured. So all those white spots are actually the clusters. Okay. So in the next seed here, and my seed, the cluster generation is on board. So the instrument will perform the cluster generation process for you. But if it's in the high seed, then we need another instrument called seedboard to do the cluster generation process. So what we do is try to amplify the signals so that we can capture it. If we don't do this step, you can imagine that the requirements for the optics will be very high. So it would be very expensive to do that. And these are different flow cells for these different instruments. So this is the flow cell for NextSeed, which is the biggest among all those instruments that we are currently offering.
So this is a single link. So for next sequence, it's only single link, not like the high seed.
For the flow cell, we have 4 links.
Okay.
Yeah. But unlike high seed, so for high seed flow cell, we have 8 links, and we can have individual samples cluster on individual links. But for NextSeed, even though we have 4 links, but since the samples are evenly distributed, so you must index your samples before putting.
So in case I want to multiplex my samples, so I need to add index rather than I can separate egg sample into egg link in high seed.
Yeah, correct. Okay, so these coming few slides will describe the whole process of the cluster generation. So before starting, we have to denature the libraries to make it single-stranded. And what we recommend is to use chemical denaturation, to use sodium hydroxide to denature the libraries. And after loaded the denatured libraries, the first step is that the single-stranded library will bind to the flow cell, and then it will do a extension. So this is the original strand, and this is the newly synthesized strand. And then you will discard the original strand, and then you will flip over to the complementary oligos adjacent and do a bridge amplification. Okay? After doing the bridge amplification, it will linearize and repeat the amplification again for thousands of times, and we'll have around 5,000 copies in one single cluster. Right? So after the amplification, it will linearize again, and then we'll cleave the reverse strand array. So what left over is the original strand. Okay? So by this way, we have already amplified the original template into a clone, what we call a cluster. And then we will plot the ends of the primers, oligos, as well as the templates to avoid unwanted priming. Is because the next step we are going to bind the sequencing primer. Okay, so we'll have it plot, and then the final step is to bind the read-one sequencing primer. So after the read-one sequencing primer is binded to the cluster, and the whole cluster generation process is completed. So any questions so far? So this is just highlighting when once you have loaded the single-stranded templates, how the cluster generation process comes across. So it's just a bridge amplification to amplify the sequence, and then finally we read one sequencing primer that contain one mutation, right? So after the sequencing primer is binded, then the next step we can go for the sequencing. And actually, there is nothing that you need to do anymore after you already learn the samples, because the instrument will suck up all the reagents and primers needed from the reagent cartridge. Okay, so this is what we do called a sequencing by synthesis from our technology. So from each cycle, we will have ATGC or bases being freshened into the instrument. So some systems will use one cycle is one single nucleotide, but from our technology, we have one cycle, all 4 nucleotides being freshened, and there will only the complementary nucleotide will be incorporated. And after the incorporation, we will image the nucleotide. That's because we have modified the molecule of the DNTP. We have a 4-pore that being incorporated into the DNTP, and we also have the 3' that being blocked. So why do we need to block the 3'? This is to ensure that every cycle there will be only one single nucleotide incorporated. So once it's incorporated, because the 3' end is blocked, so even say this molecule is complementary to the next nucleotide, it cannot incorporate because the 3' end is blocked. And after the imaging, the 4-pore will be cleaved away, and we will also have the 3' end unblocked here. So preparing the molecule to be ready for the next incorporation, or we call the next cycle. So you can just interpret one cycle equals to one base pair. So if you're like saying people, how many cycles are you going to sequence? So if they're saying 2 times 150, that means they're going to sequence pair n, and each n is 150 base pair. So this is what we mean by cycles. So actually the whole process is repeating depending on how many cycles, or some people say read length. So what is the read length? How long are you going to sequence? Or is talking about how many cycles or how many base pair that you are going to incorporate into your sequencing. Okay. So each 4-pore will give a different wavelength, and then we'll have the image that being captured, and then we can have the real-time analysis software incorporated in the instrument. It will call the base column. We'll have a detailed description on the primary data analysis tomorrow morning. So here, just give you a brief sample. So the sequencing length all depends on your application. So it, for the next step, the longest is 2 times 150 base pairs. So altogether 300 cycles. Okay. Any clear so far? Any questions so far? So of course you can sequence single read. Say for example, I'm just doing a RNA counting, and I don't necessarily need to do pair and sequencing, and of course you can do just one. This is just a fancy picture talking about the clusters overlapping with different colors of 4-pores. Okay, so, and then I briefly introduce the differences between NextSeq and our other instruments. So some of you may be familiar with HiSeq and MySeq. So, and our old instrument called GA. So for these instruments, we use a 4-channel SBS chemistry. So ATGC, each base will have the emission of 4 resins and different wavelengths. So all we call 4 channels. That's for NextSeq is a bit different. We use a 2-channel SBS chemistry. So 2-channel, only 2 images. So 2 colors. For this one, we have 4 colors. But for NextSeq, we only have 2 colors. So how are we going to make use of 2 colors? But to identify 4 nucleotides. So we use green for T nucleotide, and we use red color for C. And for A, it's half and half. So it's overlapping with green and red. So it's around things like orange color or both colors. And for G, it's not labeled. So what we do is to use a plotter algorithm like this. So if the base contains only red color signals, then it will be called C. And if it contains only green, then it will be called T. And if both colors, then it will be A. And finally, if it is not labeled, then it will be G. So some people will worry about this. So what if the cursor something right here, should I call it T or A? What about here? So we have a very sophisticated algorithms to filter away all those unknown or very or what we call outliers. Okay? And now we have version 2 reagents release. So with the version 2 reagents, the distance between these populations of clusters are much far away. That means the accuracy of point is much improved. Any questions so far? Any worries with the 2 color versus 4 color? No worries, because the quality is still very good, and we do compare the sequence data with other instruments that we offer, like MySeq and HiSeq. And we also have those data that we uploaded to our base case, and you can actually download the data and compare with our different instruments. Okay, so the next part I want you to introduce is pair and sequencing. So how many of you have never heard about pair and sequencing? That means all are familiar. So I can give a text. Okay, so pair and sequencing is a unique technology that we use and have a lot of advantages, such as for overcome the repetitive regions of bacteria or some genomes. And it can also help you to assemble your genomes more efficiently. So for de novo assembly or for fusion genes or splicing variants, with pair and sequencing, you can have a high chance of getting your data that you optimize versus single read sequencing. So what it do is, so we are going to sequence both the forward and the reverse strand. So the following slides will talk about how to do it. So this is the sequencing by the primer, level 1. So it's the final step of the cluster generation that this primer is bind to the template. Okay, still remember in a few slides we have just mentioned. So after binding to the sequencing primer, then it will go through the SBS, so different cycles, and then it will start sequencing. So first cycle we have the first nucleotide incorporator. So starting from this side is already your DNA insert. So start sequencing already. And then we have cycle 1, 2, 3, 4, and so on and goes on. So this is already your read 1 sequence. So what we call read 1 is always referring to sequencing of forward strand. So it starts sequencing. And then after that, it will block the 3' ends. Okay? And then we will have the original strand that being flushed away. And then it will, the newly synthesized strand flush away. And then the original strand will flip over, just like what it do in the cluster generation, because it have complementary oligosaccharides in the flow cell. It will flip over, and then it will do a amplification. And then we linearize the strands. So this is the original strand, the one that we have already sequenced. Okay. And then we are going to stretch this, so click this away. What left over is the reverse strand. So after that, we'll have the sequencing primer that bind again. And we have our block and the bind of the sequencing primer, and then we'll repeat the sequencing by synthesis step. Okay, so what we do, we sequence the read 2. So what we do is, so this is the original strand. We have the sequencing primer bind, and then we sequence the forward strand. And then you flip over, and then I do the amplification. And then linearize, original strand wash away. So this is the reverse strand, and then sequencing primer bind, and then I do the sequencing. So by this way, that means if this is your DNA insert, you'll have forward that being sequenced and reverse that being sequenced. So both ends is sequence. So what we call pair end. Okay. Clear with this pair end sequencing? So if this is clear, we are going to go for a little bit more complicated. Just what as Gordon asked, shall I index my libraries? Yes, because you cannot have individual samples that cluster physically on 4 different links in the flow cell. So you will have a mixture of different samples and cluster in the flow cell randomly. And if you want to pull samples, of course, you have to index your samples. So that after the sequencing, you can use the index sequence to demultiplex which bits belongs to which sample, right? So what you do is the instrument needs the index, it has to sequence the index of the libraries. So first of all, we go through something simpler, so single index read. So this is the library. The library contains only single index. So only one S is quite common, especially when the sample pooling is not that much. People will always use single index, say just 20 something or 10 samples. Then most people use single index. So what you do is similar. So we have the read one sequencing primer applying to the template during the cluster generation, and then we will sequence the read one. So you do cycle 1, 2, 3, 4, whatever. Okay. After the read one is sequenced, it will wash away this newly synthesized molecule, and then you will have the index one sequencing primer bind, and then we will sequence the index one. So now we have the read one reads, and then we have the index sequence that being read. And then after that, you wash away this newly synthesized molecule, and then you will do the pair end synthesis. So what is left over after the pair end turnaround is the reverse strand. So you can see from here the P7 switch. Okay? And then after that, we'll have the read 2 primer bind, and then we'll sequence the reverse strand. So by this way, we'll have 3 reads: read 1, index read 1, and read 2. Okay? People always refer for pair end sequencing. Read 1 is the forward strand reads, and read 2 is the reverse strand reads. And for index read, you can call it just index read 1 or index read 2. So if it's dual index, then we will have index read 2. Okay. So what about if I have dual index? So for some library, we will have dual index, especially for those that have you're going to pull a lot of samples. Say I'm going to pull 100 samples for an amplicon sequencing. Then, of course, you can design like this way, but that means you need to have 100 unique index. So if they say for single index is usually around 6 to 7 nucleotide on. If for this short nucleotide or short oligo, you need to get 100 or more combination. Is a little bit quite hard. So people will use the dual index sequence. So we just like a mix and match, and then we will have, say for example, sample 1 and 2, we share the same index 1, but we have different index 2. And with this way, you only need the limited combinations of index, but you have a lot of different multiplicity capacities that you can work. Right? So this is the advantage of dual indexing. So the workflow is quite similar. So I'm going to go for the basic one first. Okay. So what you do is to again bind the read 1 sequencing primer into the read 1 sequencing, wash this away, and then sequence the index read 1. Okay? And after that, you'll do the pair end turnaround. And instead of going directly to read 2, it will do the index read 2 first. And then wash this away, and then we will have the reverse of the read 2 that being sequenced. So for dual index library, we will have read 1, index read 1, index read 2, and read 2. We will have 4 reads altogether. Okay. So this is what you do for Nextseed. This is for your reference. For other instruments, It's a little bit different. So after the index read one, it will flip over and then it will go through 7 cycles, what we call chemistry only cycles, or what we call dark cycles. So it's called dark cycle because it's not image, but it still consumes your sequencing reagents. Okay? But it's already included in the reagent, so don't worry. And after the dark cycles, this aims to repair the adapters which already clipped in previous steps, and then you will sequence the index 2, and then complete the pair and turn around, and then finally sequence the root 2. So for those who will send out your samples for other instruments like the high-seq, sometimes you will calculate for the SBS reagents how many cycles you will need to consider for the pair N or U index. You have to add 7 cycles, additional cycles, right? But for mixed, you don't need those 7 cycles, additional cycles, because it's complete. After complete the pair and turn around, and then it will give the index 2. Okay, any questions so far? Okay, so what we just mentioned is the first step, after you denature your library, it will do a cluster generation to amplify the signal of the technique. And the final step of cluster generation is to bind the sequencing primary. And then depends on the design of your library. Is it a single read or pair N sequencing? Or is it the single index or dual index? It will have different reads that go through the whole process. Depends on your library design. The kits will already include all the reagents necessary for dual index pair and sequence. So finally, data analysis. So we'll have a detailed session tomorrow morning, but briefly, we'll have a control software, Connexit Control Software or NCS, and bind together with the NCS, we have an analysis software called RTA, Real-Time Analysis Viewer. So these two software actually work together. Once the Connexit Control Software started the sequencing, the RTA will start to work. So the analysis is in real time. So it will not wait until you complete all the imaging and then it launch analysis. No. So after you complete the first cycle imaging, RTA will already process the image that taken in cycle 1 and start doing the analysis. And that's why we can much reduce the sequencing time. Okay. And then with the output from the primary analysis is the base code files, BCL. And you can use our BCL to FastQ2 software to convert the BCL file to FastQ files. And have these FastQ files, you can use a lot of common bioinformatics tools to do the data visualization. So what the visualization is called, sometimes we call tertiary analysis. So this slide highlights all the differences of the 3 different stages of analysis. So the first stage comes with the control software. The Connexit control software, or in my seed is MCS, or high-seq is HCS, that bind together with the analysis software called RTA. So what you do is to convert. So the NCS will capture the raw image of the clusters for different signals. Is green, red, both color, or is black. And then the RTA will convert the image into a base code. And that's why after the sequencing, what you get from the instrument is the base code files. So, but with only the base code files, it's really hard for human being eyes to interpret because it's just saying that for this cluster, this cycle is which nucleotide. But what we want to do is a sequence of that. Okay. So what you do, you can use either BCL to FastQ2 software, which is a Nimbus supported software. With this software, you can convert the base code files into alignments or FASTQ files, or variant files, or what we call VCF, variant code files. I have these files, FASTQ, variant code, BAM files. You can use these as input and then make use of other tools or the apps in the base space, and then you can convert it to annotation files. So that means even though I have the variants, but are the variants meaningful to me? Is it a common polymorphism? Is it really a pathogenic? So what you do need to annotate. So annotate is to compare your own data with known database, and to see whether there are any matches in the database, and then you can go further for interpretations. Okay, so these are the three different stages of data analysis in NGS. Right, so any questions so far for this part? Okay. So how many of you are doing dual index sequencing? One. So the rest of you are single index. So that means, so how many samples are you?
Usually 16.
16. That's usually used. Right. So no problem. No questions. So what are the major applications in the teams? I know some of you are interested in exome sequencing, in RNA sequencing, any other applications that you guys are interested? No? So basically RNA-seq and exome. Okay. So what the
Whole genome sequence.
whole genome sequencing. Okay. So good news is in next week we can also do whole genome sequencing for human genome. Yeah, you can do one sample for one whole genome sequencing. So the NextSeq is the most flexible platform available. MySeq is more suitable for targeted sequencing. If you want to do like whole exome sequencing, whole human transcriptome or whole genome sequencing, the throughput is not high enough. And for HiSeq, because the throughput is very high, so if you have limited samples, then usually you need to wait and do a lot of samples in one single run. So NextSeq is a little bit flexible in that way. Right, so if no questions, then we can proceed for the next one. So this session will have a brief introduction to the NextSeq system. So for NextSeq, we have two models. One is called NextSeq 500. That's the one that we purchased in our institute. And another one is called NextSeq 550. So the main difference is that 550 with a scanning function for our Infinium array as well. So good news is that you can upgrade anytime if you think, say, for in future you are interested in doing Infinium microarray, and you can upgrade your NextSeq 500 to NextSeq 550. But for the sequencing part, they're basically the same. So this part, we are going to identify the features of NextSeq system, what are the consumables, and describe about the sequencing technology, which I think I have already touched in the last section. So NextSeq 500 supports exome sequencing, whole transcriptome sequencing, and whole genome sequencing. And as I just mentioned, it has a very flexible sample size, and we have two different full cell options, what we call mid output or high output flow cells. So depending on the throughput that required, you can choose among mid or high output. And you can scale from single sample to thousands of samples per run. And the design is low and low. So after you have already loaded your samples and started the run, then you can just leave it and then after the completion, you come back to get your base core files. Okay. And you can also connect your Nexus system to our base, which is a cloud-based server. And with that, if it is connected, this data will be uploaded to the cloud. And after the completion, you can simply launch the apps to do the analysis in the cloud. And if your run is connected to the cloud and you have assigned different samples with the index, the cloud will do the multiplexing and FastQ generation for you as well. So may I know, is the instrument now connected to internet already? Is it connected to internet now? Oh, okay. So if you want, we can try if we can have it connected, and then we can still have the run uploaded to the bases. Okay. If say you have concerns, I have security concerns, I don't want my data to put in the cloud, what can I do? You can do standalone run. So you keep all those data either on instrument, but we recommend a network drive because the data size is quite big. You can do a standalone run, have all those PCL files saved in your network drive, and then you can process the data with the PCL to FastQ, which is the Linux supported software, and then you can still do the multiplexing and the FastQ files generation.
I save the data in the network drive and I still use the cloud software to do that.
No, if you store your network drive, then the output file will be PCL file. But after you have converted PCL to FastQ by the Linux supported PCL to FastQ, you can upload the FastQ to the cloud and launch the apps for further analysis.
Is there a storage limit in space?
Yes, we have storage limit of 1 terabyte for free. Yeah.
Which is per person.
Per person, yes,
So
per
if
account.
the run exceeds that,
You can delete your old files and spare extra. Yeah, but I heard from our marketing that we are going to charge for the computing starting April this year.
so place.
Yeah, you can say that, but I don't have any very solid details on how much it's going to charge, but I heard that we are not going to earn money from that. We're just to cover all those that we pay for Amazon's so
What is the storage? No,
storage is still 1 terabyte free.
the software is.
But if you like the computing on the base, you will start to charge. But honestly, I don't know how much it's going to charge you.
So as you mentioned, we don't need to do the analysis on the cloud.
You can either do it in the cloud or do it by yourself. As far as you have a Linux supported computer, you can install the software called PCL to FastQ2, which is free. So you can download the software by free from our website, and then you can convert the base code files to the FastQ files. So is there any?
Do you think we have the chance to get your free software or hardware? So before we want to, you haven't run your software, how do you know it's
You mean the PCL to FastQ is free?
But I want to know it is in the local region.
Yeah, you can do it in your local drive. So say you have so forget about all those base days. You just save the data either on local drive or your network drive. What you do, you have to copy your run data or the base code files, and then you generate a sample sheet. So how you generate a sample sheet, you can use our free software called IEM. You generate the sample sheet basically states your sample number, your sample name, and the index used for that sample. It's just like a map for the software to grab which file, so to do the multiplexing. So what you do is your run data, a Linux computer install with the free PCL to FastQ files, and you prepare a sample sheet, and then you launch the software, and then you will convert the base code into FastQ files.
That's me, you
Yes, sure. Linux.
How about the analysis software?
It's free.
Also free.
It's free. Yeah.
So why the house? Why if we got the server, why we charge the house?
If you do, so it's just an alternative option for people who want to use the cloud. Because in the cloud, the advantage is that it's not only doing the demultiplication and FastQ generation. With the final FastQ files, you can launch the apps in the cloud. Like for RNA sequencing, used with an Illumina with the top hat and coupling analysis algorithm, or for exon, maybe you want to use the BWA enrichment or ISA enrichment workflow for, it's more like secondary annotations analysis, all those.
So that's analysis. Do we get any chance to try that analysis
in your local server?
Yeah.
Local server, no, because the app is in the cloud server. But
no local software for this app.
No local one, unless you use the base on-site. So the base on-site is a server, but the server with inbuilt apps like what we have in the cloud, but it's not collectively internet. You can ask a phone.
So can we try this one?
Try? I don't think you can try, but if you want to say you want to play around with the apps, because now it's still free, right? So after completion of this one, you can already have your FastQ files and then you can launch the apps and then control.
So because set up a unit server is not easy. Before we buy the so-called this app now, how come I know this type of Linux app?
Well, to set up Linux is basically aims to convert the PCL to FastQ, but it's not we're not providing the tertiary analysis like apps in the based innovation. So it all depends on it. So we have all those pros and cons for different options.
Is the conversion from the computer ABC to FastQ?
Yes, you have
So
to
it's just basically.
What? Any more questions?
Where do you do the demultiplexing?
Where do we do the?
In which steps? Or do you get a demultiplex PCL done?
Yes. You will, no, you will do the demultiplex FastQ after the conversion. So after the sequencing, it will not be demultiplex. It will only after you convert it with either base or the PCL to FastQ software, it will be demultiplex, and then what you get is a demultiplexed FastQ files.
So is the index stored with the read language as you get to?
The index will have an index with one and index. The index will find in the FastQ files.
With each.
Yeah. Any more questions? Right, thanks a lot for the good questions. Alright, so this is the two different flow cells from the Nixie system. This is the high output flow cell, which supports 400 millions of reads and up to 120 gigs of output. And the mid output flow cell is basically 1/3 output of the high output flow cell. You can just by NECA you can tell the difference. So this is the name of the flow cell 1, 2, 3, 4. And this again, so for the mid output, it also contains 4 lanes, but you can see that the width of the lanes is narrower than the high output. Okay, I will talk about that in a minute. So, and this is only 1/3 of that. I guess you're familiar with the output and reads, right? Okay, so for RNA person, usually they look for number of reads available for each sample. But for DNA people, they will calculate the coverage, and that's why they need to know the total output. So it's a basic calculation. With the reads times the read length, then is the output. Right. So if you are talking about the coverage for DNA sequencing or whole genome sequencing, say you want to have 30x coverage, then you need to calculate. So the total output, if you sequence pair N150, you will get 120 gigabytes of data. And then you have to calculate the coverage. So you use the coverage times your sample's genome size or the target size. This is what you need, the output per sample. And then you just divide the total available output from the flow cell with the total output per cycle that you need, then you will know how many cycles you can do in one single run. Okay? Don't worry, we have a coverage calculator in our website, so anytime you can play around with the website. It will calculate for different flow cells, different lengths, and then you will have an idea on how many cycles you can do in one single run.
Sorry, but for mixed, is it only pair end flow cell available, right?
Yes.
So if I'm doing the RNA sequence, I only have to choose the pair end chips.
The flow cell is pair end that you can do single read.
And the time is also double, is it?
The time will be shorter.
So I can choose to terminate the run in the middle of the
No,
sequence.
you don't terminate the run, but during the run set, I will tell you that it's a single read, and you will stop.
And then I waste another half of the flow cell, another half of my run.
You can say that. You can say that. So this is the performance or the specification of the Maxi reagents. So you can see we have two different types. The high output flow cell, we have three options. 1 times 75 cycles, 150, so it's 2 times 75 cycles or 2 times 150. So it depends how long you're going to sequence for your libraries. Say if I'm just doing a simple RNA counting, probably 1 times 75. Maybe some people usually do around 36 to 50 base pair for counting. Then this you don't need to purchase a 300 cycles kit. Maybe 75 cycles is good enough. Okay. And you can see the sequencing time is different as well, because for shorter sequencing, the imaging time will be much reduced. So it only takes around 11 hours to complete a single read 75 cycle sequencing. But if we are doing to 2 times 150, then it takes 29 hours. And for the mid output, we don't have 1 times 75 cycles option. So unfortunately, if like you have a few samples and you still want to do 50 cycles of single read sequencing, then you still need this kit. But that means that you are going to waste almost more than half of the sequencing images. Alright, so this is the data quality specification. We use the Q30, which means that, say for this one, we guarantee over 70% of base are more than Q30. So Q30 is a quality matrix talking about what the probability of calling one base wrong out of 1,000 base pair, which means you're talking about 99.9% of accuracy. So we guarantee over 70% of base with accuracy of 99.9% for a 2 times 150 base run. If it is shorter, say 2 times 75, this is a lot of the choices for RNA sequencing, 2 times 25, then we guarantee over 80% of your base about Q30. Okay? And same as for the output. And just remind that the recommended cluster density on the mixing system is between 170 and 230 K per mm squared. So this is much different from what you get from the high seed or my seed, which is near 1,000 K per mm squared. It's because the design of the flow cell is different. Yeah, but not essentially meaning that you have lower cluster density than you have lower output. No, because the flow cell design is completely different. For my seed, the flow cell is just like this size, but for mixed seed, it's this size. So it's different. So cannot directly compare with these with other instruments. But just remember this, because the cluster density is going to affect your yield as well as your data quality. Okay? I will talk about that in tomorrow. Why is it library quality and quantitation so important for you to ensure that a run can be completed with a successful and good data quality. Okay, so this is what I just mentioned in previous section. So it contains 4 lanes, 1, 2, 3, 4. But the library is flows through the 4 links. Okay, so unlike the high-seq, because we have the C-board, and the C-board will suck up each routes of library and physically cluster the libraries in different links. But for Mixit, no, because we only have 1 lines to suck up the mixed libraries or the pool of libraries. So you will suck up the libraries, and then you will randomly and evenly distribute it across the 4 links. And that means there is no way that you can unindex your samples, but pool them and pull through the Mixit and expect it to do the multiplexing. No, you must index your samples before you can do that. Okay? And hybridization, cluster generation are automated on the system, and approximately there will be around 5,000 of molecules in one single cluster. Okay? This is the 2-channel chemistry. As just mentioned. I think I've proved this already. So I just want to point out here that because the G is unlabeled, is black color. So some users will ask me, so how is the instrument going to identify is really background or is a G is a cluster, but it's a G nucleotide. It's because during the first 5 cycles of the template generation, what we call the first 5 cycle is very important for any instrument, any of our instrument, because it uses the first 5 cycles to generate what we call template. So a template generation is essentially try to identify the exact location of the cluster. So you can imagine, since you randomly fetching the libraries, and there is a possibility that 2 clusters could be very close together or even partially overlapping, right? If I'm only use this, say the first cycles to tell whether this is A or is 2 or is only 1 single cluster, it's very risky, because there's a chance that there are 2 different clusters, but they are actually essentially sharing the same base at that particular cycle. And if I use that only 1 single cycle to identify or to try to separate these 2 clusters away is not possible. So we use 5 cycles to do that. So unless your libraries in the first 5 cycles they are all G, then the cluster will be filtered away, because the instrument will recognize that as a background instead of a cluster. But what we know is that from our genome we have consecutive 5G is not that high, and that's why only G is is not labeled instead of using other nucleotides, right? And because also have this issue, we recommend users not even in the index read try to avoid index that with first 2 cycles that are G. So some index with GG and then other sequence. So avoid with, so we need at least 1 non-G in the first 2 bases of the index. And if you're using Illumina's index, there is only 1 index that starts with GG. But I know some of you might be using the third-party kits. So before that, please review your kit index to avoid index start with GG. Right. I have some users tell me that I tried that, it works, but officially it's not recommended to use index with GG. Okay. So this is so the main benefits of using 2-die chemistry instead of 4-channel chemistry is to reduce the imaging time. So if I'm using the 4th channel, I have to capture the 4 different fluorescence signals. But for this one, I only capture 2 colors, and I use these algorithms to determine the phase. So it will reduce half, 50% of the imaging time. So what you do is try to increase the turnaround, so to reduce the turnaround time. So these are also mentioned already. So next, let me briefly introduce the hardware. So this is the most expensive part of the instrument, is the optics. The optics is designed in a solid state, so it has 6 cameras. So people ask me, you have only 2 channels, then why do we need 6 cameras? It's because you want to take image for different sections of the process simultaneously. So the aim is also to reduce the imaging time. Right? What you do is to have the 6 cameras all pre-passed in the stage. So what we call solid states. And the advantage is that low alignment is required. Like for high seed and mixed seed, we have lots of optics, mirrors, filters in the instruments, and we need a lot of alignment during the installation. But for mixed seed, because it's already pre-installed, so no alignment is required. But having said that, it doesn't mean that you can move the instrument anytime you want. No, because if the whole stage is altered, it will still affect the accuracy of the image acquisition. So just leave the instrument right there. If you want to relocate the instrument, please contact us. Yeah, we need our engineer on site to lock some of the areas inside the instrument. We need a special trained person to relocate the instrument, and that's a charge. Okay, so and do not put centrifuge or vortex around the instrument. So it's very sensitive to vibrations. So just take note of that. Okay, so this is optics, and that also means that if single part of the optics has problems, there is no way that we can say the camera 1 has issues. We can not only replace camera 1, we need to replace the whole modules of the optics. But I know you have 1 year's warranty, so don't worry if it's within warranty. But we don't have a lot of issues with that. So the 6 cameras will take image simultaneously. So this will allows it to have a higher throughput and keeping a very short turnaround time. So the runtime is very short. It has 6 times imaging capacity compared with the MySIF, and it only have 1/3 the size of the high-SIF system, and the cost is also cheaper. Right? So having a lot of advantage. And this is the MySIF Vortex system again, and is all pre-cast in a manifold, Vortex manifolds like this. So these are the zippers. The zippers are actually, it will move down and then punch through the reagent cartridge, and then the reagents will be passing through from the zippers. And then here you can see there are some lines, the Vortex. And then you will go for the Vortex and then go for the process. The major breakthrough of this design is to reduce the tubings. You can see the lines here is very short. The distance is very short. That means the depth volume inside is much reduced compared with other systems, and you can lower the price for the reagents. And also one major is that with a very limited tubings, you can lower the maintenance steps. So in high-C, if some of you have experience, basically you have to wash the system every day because there are a lot of long lines inside the instrument, and you have to make sure that there is no blocks or no leftover chemicals or precipitates inside the instrument. So basically you need to wash it every day. But for the next seat, because of the limited design of the tubings, basically you only need limited efforts to maintain the instrument. So after every sequencing, it will automatically perform its post-run wash. And you only need to manually wash the instrument every 2 weeks. And that's all for routine maintenance. It's very simple. Right, and then this is the consumables of the Nexi system. Basically, three major parts. So this is the flow cell. Flow cell is should be storage of 4 degrees. Never freeze the flow cell. Okay? If you accidentally freeze the flow cell, please do not use it. It's not going to work. And then this is the reagent cartridge. The reagent cartridge contains all those polymerase, primers, and all necessary buffers required for the SBS chemistry and the custard generation process. And what you need to do, you can see the orange well here. This is the well where you pipette your denatured and diluted samples, just like the mice. What you need to do is to load the diluted samples, then that's all. There is nothing else that you need to know unless you're doing custom primer sequencing. So say if you're using third-party library packets to do the sequencing, and they are not using the compatible sequencing primer is what we provide in the cartridge, then we also have spare wells where you can put the custom sequencing primer. So before sequencing, make sure that your library or the third-party kit that you're using does not require custom primer. If you do, you have to prepare the custom sequencing primer. Otherwise, you will not able to sequence it because the sequencing primer is, if it's not compatible, of course, the primer cannot bind, and you are not going to get any signals from sequencing. Okay, so these three graded wells are for the custom sequencing primers, and we also have. And this cartridge has to be stored at -20, and it could be quite bulky upon arrival, a big box like this. Okay, so if you are going to order quite a number of the cartridge, make sure you have enough fridge space to hold the reagents. And this is the buffer tray. So the buffer tray contains some incorporation buffer and the watch buffer for the post-run watches. Okay, so the consumables, you can see that this lists the actual number of cycles that we use supplied for each kit. So for example, for 70 cycles, it's not only provides 75 cycles, it actually contains 93 cycles. And for 150, it contains 168, and 300, it contains 380 cycles. So why is it so generous to give few more cycles? Is because what we call like 30 cycles is supposed to times 150 base pair of sequencing reagents for your insert. So if it provides 2 times 150, then you can make use of the whole reagents to sequence your insert. And the extra cycles are required for the index reads. So say 18 cycles, actually it provides the 8 cycles for index read 1 and 8 cycles will be index read 2. That's 16 cycles. And the extra 2 cycles is for phasing and pre-phasing calculation. I will talk about that in tomorrow's session. Basically, is say if your insert you want to sequence 2 times 150, then I want you to input as 2 times 151. I want one single molecule, one more cycles for each read. That's for phasing pre-phasing calculation. And that 2 cycles is included as well. And that's why it's 300 times 8 times 8, so 2 index reads, and then times 6, 2 more cycles of phasing pre-phasing. And that also means, say, if I'm doing only single index, and that means you have 8 extra cycles left over, and you can, if you want, you can say make use of the 8 cycles. I want to 2 times 155. Yeah, you can use that. Okay, and as I just mentioned, the reagent cartridge storage is -20. Buffer tray is room temperature, and the flow cell is 4 degrees. And it will also come along with a accessory box, which contains only a buffer called hybridization buffer HT1. So the buffer is required for diluting your libraries after the denaturation. So also at -20. And the buffer trait is universal, meaning that for either high output or mid output kits, the buffer trait is essentially the same. But for cartridge and flow cell, it will have a label telling that it's high output or mid output. So if say you will have both different kits in your lab, make sure that you use the right flow cell and the right cartridge. Okay. The instrument will check for the compatibility. Say for example, you load a mid output flow cell, but you use a high output cartridge, it will go on you tell you that you are not using the right one. But it will not check the number of cycles. I do come across with a case that will run in week 2, the intensity drops suddenly, and eventually the root cause is that the customer sequence of 2 times 150 cycles run, but it used 150 cycles reagent cartridge. And that means in week 2 somewhere, it already consumed all those reagents and then intensity drops suddenly. So make sure you use the correct kit and the correct cycles for your sequencing. Okay, and for the flow cell and the cartridge, we will have label, either high or mid. But it's always a good practice to check by yourself as well every time. Okay, so this is the flow cell. The area that are going to cluster and imaging is these 4 lanes gray area. But never touch this area with your bare hand or even when you wear your gloves. So we have a handle, the right size of the handles where you can handle the flow cell. So what we suggest is to wipe this way, just like all the flow cell like this instead of this. And the flow cell is shipped dry. So unlike high seed and my seed is shipped in a high sulfur and you need a lot of rinse before use. But for mix is much improved, it's shipped dry. You just need briefly use ethanol to clean it, to wipe away any like dust or things, that's all. Okay. And we have a color icons showing its high output or mid output flow cell. And it also has RFID chips in the flow cell. So the instrument will detect the flow cell with the RFID to see it's compatible with the one setup that you want. So, and it's a dual surface flow cell. So what we mean by dual surface, imagine in a flow cell, if I cut it over, so we have a plastic in the upper layer and the lower layer, right? And the fluids are going to flow through in the middle. And the cluster actually form from here, the top surface. So we'll have the cluster here, but we will also have cluster in the bottom surface. And that's what we call dual surface. So we have 2 surfaces that contains cluster. The aim is to increase the sequencing throughput. So both surfaces will make use of it. Both surfaces will have cluster, and the SBS process will run in both surfaces. Okay. And we have 4 lanes, and again, 1 sample or 1 pool is distributed across all 4 lanes. And subdivided to that, we have call lane 1 and 3 as lane pair A, and lane 2 and 4 as lane pair B. Okay, and the flow is like that. For lane pair A, it will flow through from up to the down, and lane pair B is from down to the top. And why do we need to separate into lane pair A and B is because the 6 cameras and the chemistry is doing their job simultaneously. So if we magnify the flow cell like this, we can subdivide the flow cell into 6 camera sections. Section 1, 2, 3, 4, 5, 6. Okay. And each section, you can subdivide it and each lane. So this is only lane 1 and lane 2. Okay, from here. And each lane will subdivide the areas into 3 swaps. Swap 1, 2, 3, and each swap will subdivide into 12 tiles. Tile 1 to tile 12. So for the imaging, even though we have 6 cameras, it doesn't mean that each camera takes the picture from this area. No, it's taking pictures from one single small tiles. So very precise because the cuts are very small in microns, right? So it subdivide and then it will, so camera 1, 2, 3, it's imaging, and so 6 camera will image together. So that means link pair A will be imaged at the same time. Camera 1, 2, 3, 4, 5, 6, the ticket imaging. But link pair B, so if link pair A is doing imaging, then link pair B will be doing the chemistry, and vice versa. And again, the main advantage is to reduce the sequencing time. Okay, right. So, and then each image or each base code files that you will get, it will contain a long number. So this number is not random; it's designed. So the first one, 2, is the name. So this is name 2, and then the and the underscore, and then this one is either is top surface is 1, bottom surface is 2, and then the swap numbers, camera section number, and the tile number. So for example, if I have this number S121304 red, so what does it mean? That means this is the red channel image, and is, so the first one is name, right? So it's name 1, so name 1, and it's bottom surface because it's 2, and then 1 is swap 1, so name 1, swap 1, and then camera 3, so somewhere here, and tile 4, it should be somewhere here. Okay, but it's just for your reference. You don't essentially need to like grab all those data, because in the B-cell to FASTQ conversion, it will like automatically convert the files to FASTQ. Yeah, just automatically. Okay. And this is the reagent cartridge. As I just mentioned, it's preloaded with single-use reagents, so it's single-use only. It's a consumable, not reusable, and contains all reagents required for cluster generation, sequencing, and pair-end chemistry. And single library or pool of index library can be loaded in the cartridge in this well, so number 10. And each well will have a corresponding empty well adjacent. So when you look at the reagent cartridge, you can see that these blue wells are empty. That's normal because they are reserved for the wash. So after completion of the run, the zipper will pump the wash reagents from the reagent buffer tray to the empty wells because they're clean, right? They're empty. And then the zipper will move to the corresponding. So for number 6, the zipper will move to 1 to pump the wash buffer and then wash the lines. And for number 28 is bleach, so it will wash a template line as well. So some people worry about what about the percentage of carryover. So what we mean carryover is say the first one I have index 1, 2, 3, and I sequence, even though you say you have wash, but is it possible there is still some library left over, and then if my next one I also use index 1, 2, 3, is it going to have some previous runs libraries that contaminate my this run's data? So the carryover rate is normal for each instrument, but the percentage in general very low. It's lower than 0.1%. But because of this concern, we incorporate a bleach wash to wash the template line. So already like wash away all those templates left over the last one. But if there's a still concern, some people even play it more safe. Say for this one I use index 1, 2, 3. Maybe next one I don't use this index, I use another index. Then you can completely like eliminate all those possibility of carryover. And for the reagent buffer, actually it have 3 wells. We have Number one is the sequencing buffer, and then we have watch buffer here, and the incorporation buffer. They are all preloaded, so there's nothing here. And the instrument is quite small actually, and you can see this is a pre-built computer, and it's touch screen, so you can just use your finger and touch, and then ask it to go through the sequencing. And we have a main switch at the back, in the left upper panel at the back, and this is the main switch for computer. And here is, so the flow cell is going to load it in here. Here is the flow cell cartridge. And then here is the reagent cartridge to fit in the reagent tray. And the upper section here is for the reagent buffer. And this is the waste container it's going to put in here. Okay. Don't worry, we'll have a hands-on session this afternoon to practice. And so all those are the hardwares, and what are the software? We have the Nexus control software. So the NCS will guide you through all the steps to load consumables and to start the run. So, like, even though I'm not here today, I'm sure you're able to start your own run. Because it's really easy, and there is a videos and tabs telling you which tray and which buffer to know. So that I don't see any problems you might encounter. So it's very user friendly. And as I just mentioned, for primary analysis, we use NCS and RTA2, and it's an on-instrument computer that will all compute primary analysis. And because the RTA will run in memory, so after it converts all those intensity data, all those image into intensity data, it will not save the intensity data itself. And that means you need to spare some hardware space for the RTA to run. And we don't recommend store more than one run on the instrument. And that's why we suggest use either network drive or you can interface. Okay? And for data output, so after starting to run, the NCF will generate a run folder. And I think we can have a chance to have a look at the run folder tomorrow in the data review section. So it will contain some base programs. And the thumbnail image, the thumbnail image is the actual image that the instrument captured. So just like the one with black background and there's some white spots, right? Those are thumbnail images. By default, the thumbnail image is not saved because it's going to occupy quite a lot of the space of the hard disk. But personally, I suggest you to turn it on, especially when you are at the very beginning to start your own run. So because the thumbnails image are very useful for troubleshooting purposes. Say for example, over clustering or under clustering, if there is no thumbnails, then it's a bit hard to really determine whether this is the root cause. So if you agree, I can try to turn the save thumbnail image on if you want. Yeah. Okay, so and the data generated from the instrument is compressed. So it's 6 ECL files and with bin Q scores. But don't worry, the ECL to FASTQ file is compatible with zip ECL files. So you don't need to unzip every cycle. That's quite tedious. So it's compatible. And for the multiplexing, you can use the BCL to FASTQ version 2 software. Okay. Again, that is no instrument FASTQ version. You need this software with a Linux supported computer, or you can connect to base for the multiplexing. So this is the base. It's a cloud-based server, and you can choose upload the one quality matrix data only or the whole run. If you choose upload the whole run, then you can have the multiplexing and the FASTQ generation. And if you just say, for example, I don't want to disclose my data, and you can choose just upload the one quality matrix in the base. And in that way, if there is some issue, you can share the one with me, and I can monitor more images. We will have a detailed session on the base space apps tomorrow. So I'll leave it to tomorrow. So for the workflow, so this is just a brief screenshot of what people usually do with Mixit. So for RNA gene expression profiling, and say if you're going to have 10 million of this per sample, you can run up to 40 samples per run. This only does not mean that you cannot do it in mid-output. It's just because we didn't test it. But of course, it's compatible. Because the output of mid-output is 1/3 of this. So that means you can run 12 samples with the mid-output flow cell. And you can do transcription sequencing, enrichment sequencing. So I guess this is the 271 panel. And whole exome sequencing, you can do up to 9 samples. And for whole genome, human whole genome sequencing, you need 1 sample. So some people also ask me, what if I have a sample, I want to do whole genome sequencing, but the genome is larger than human genome. Can I still do it on Mixit? Yes, you can still do on it, but you might need more than 1 runs of data. Say for example, the genome is 3 gigabytes. You can do 2 runs, and then you can pull the samples together for analysis. Yeah, that's another way that you can do it. So the whole workflow on preparing the libraries to sequence on the Mixit is very simple. I guess for beginners, it might take you around half an hour maximum to prepare the libraries before sequencing. It's just a simple denaturation, neutralization, and then a dilution. And then you load it in the cartridge, and then you can go to the instrument and start the sequencing. I guess it takes around 20 minutes. And after that, you will analyze to get to the BCL files. And if it's connected to base space, you will have to pass to. And say if you have connected the run in the base space, then you can also share your run with your colleagues or your collaborators. Say for example, I have these samples on base space, and I have played around with many apps, and that means I have some analyzed data. You can actually share the whole project. That means you will have the raw FASTQ files as well as the analyzed data in the project, and your collaborator will have all those once you have shared. So that's another benefit of having a run connected to the base space. Okay. So any questions so far?
Conceptually, when you say there are 2 surfaces, how does the camera know which surfaces?
It will do is autofocus actually. It will do the autofocus for the top surface. Of course, we have a specification to the installation time, approximately which area. And then after capture the top surface, it will move down because we know the exact distance between the top surface and the bottom surface. And then it will move down to see the pictures of the bottom surface.
So the clusters are not overlapped. So you won't capture the top.
No, they are not overlapped. Yeah. And you can tell. So that's a very good question. And if you have the thumbnail image that being stored, you will know. If say for unknown reason is out of focus, and then you have the thumbnail image, you can tell. Say there is no image in the top surface, or something is out of focus, then you can tell all those differences. Any questions? If no, I guess we can take a break. Maybe 15 minutes or 20 minutes. We'll come back at 11:20. I'll be here, so if you have any questions, please come across.